Good morning, everybody. Thank you so much for joining us here. This morning, we are super excited to kick off the first annual symposium of our Center for Cancer Systems Biology, which is a NCI-funded center joint between MIT, Dana-Farber, and a few other institutions that we will meet today during our morning talks. My name is Franziska Micha. I'm a professor at the Dana-Farber Cancer Institute at Harvard and have the great pleasure to co-lead the center with Forrest, who will also be our first speaker and introduce us to the overarching goal of the center, as well as his research. Forrest holds a bachelor's degree in chemistry from Framingham State College and a PhD in analytical chemistry from Florida State. He then did a postdoc at the University of Virginia and left for industry, but luckily for us, came back and has been at MIT since 2003, where he is now a professor of biological engineering. And the only downside is that he is not an Antonio, because all others in the morning session are an Antonio, so he is an honorary Antonio for this morning. And we very much look forward to your talk.
Thanks. All right. Yeah, so thank you all for coming this morning. I know it's kind of early, but I'll get started with a quick overview of our center. So I won't go through the whole name because it's rather long. I think we just decided to throw everything together into a single name, but we have really two projects, and the center is really focused on understanding the interactions between GBM tumor cells and neurons, as well as GBM tumor cells and immune cells in the tumor microenvironment. And then ideally, sort of bringing all of this information together to try and understand the interaction and the dynamic evolution of GBM tumor with its microenvironment, including the neurons and immune cells. The center is composed of Franziska and I as the co-PIs, and then we have some awesome, talented people, including multiple of the people you're going to hear from today, as our co-investigators in our center, covering a range of different topics. I'd also like to just give a quick shout out to the National Cancer Institute. So Hannah, Monica, and Shannon are the program directors and deputy director for the Division of Cancer Biology. And they've been fantastic supporters, as well as giving us guidance for our program. Most importantly, I'd like to call out, give a quick shout out to Isadora, who's sitting in the back so she doesn't get embarrassed. I think in the other room maybe, but anyway, Isadora put everything together today and tomorrow for our site visit for NCI, and has just been phenomenal at getting everything set up. Okay, so I won't belabor an introduction to GBM, but just quickly, it's one of the most common and most deadly form of adult brain cancer. Survival is relatively limited, and treatment is really focused on resection, radiation, and chemotherapy. And unfortunately, as you'll hear probably a couple of more times today, that treatment paradigm really hasn't changed much over the last 20 or 30 years. And there's been just dozens of failed clinical trials in this disease. And so a couple of the challenges that I'm going to sort of hit on today, one of them is the invasive disseminated cells. So these are the cells from the GBM tumor that crawl into the normal brain and invade the normal brain, and not only the brain around the tumor, but also evade across the hemisphere to the other side of the brain. And these disseminated cells basically drive recurrence after surgery, and often lead to that recurrence being fatal. And so what we'd like to be able to do is to actually characterize and target these disseminated cells that lead to recurrence if we can. But it's been super challenging because identifying and isolating these disseminated cells when they're hidden behind billions of neurons is really, really challenging. The other very challenging aspect of GBM is that it's incredibly immunosuppressive. It's a cold tumor, meaning that there's not a whole lot of mutations present, and there's minimal T-cell infiltration into the tumor. But a lot of this is driven by these glioma-associated macrophages and microglia, the GAMs. And these GAMs basically drive this immunosuppressive phenotype, secrete cytokines that drive away T cells, and really limit the efficacy of immunotherapy in GBM. So we'd like to be able to tackle these. I'll talk about these sort of in order today. So the first one is the invasive and disseminated cells. And so if we look at a couple of tumors, GBM6 and GBM12, these are PDX tumors, patient-derived xenograft tumors grown in mice. If we look at the opposite hemisphere, you can see there are little tumor cells that have crawled over and have invaded that opposite hemisphere and are hidden behind, like I said, the neurons and normal brain tissue. And in GBM6, if we look at this region here, we get about 5% of the cells that might be tumor cells. In GBM12, we have a very small amount of cells that are tumor cells that have invaded through. And the challenge really is isolating and characterizing these cells, ideally while preserving their physiological signaling state. So we can't just dissociate the tissue because that changes the cells as we dissociate the tissue. So what we need to do is find some way of sort of fixing that state of the cells in order to isolate them and identify them. And so Yejin An, one of my former postdocs in the lab, recently developed this technology that she's calling Insight. And it's a way of basically being able to isolate and characterize particular cell types from tissues for downstream phosphoproteomic analysis. And so the way that this works is we take out the frozen brain. So we take out the brain, freeze it. We can then cut it into hemispheres if we want. We can section those hemispheres on a cryostat and then drop these sections into formalin to fix them. And this basically preserves the physiological state of those cells because they've been frozen or fixed the entire time. We can then dissociate them into single cells and then sort by cell types on a cell sorter to isolate out particular cell types that we're interested in. We could sort by CD45 positive cells to get the immune component. We can sort by EGFR status to get the tumors. We can then lyse those cells and then basically process them through our standard mass spec protocol to enrich for phosphopeptides and then quantify phosphopeptides from these various cell types. And then at the end, we get basically peptide identification, including the phosphorylation site on the given protein, and then quantification as to how much it changes across the different samples that we might be interested in. All right, so Yejin applied this to GBM6 and GBM12 in collaboration with Jan Sarkaria's lab, where they injected GBM6 or GBM12 into the right hemisphere, wait 24 to 28 days, and then basically take out the tumor and take out the brain and freeze it. And then we cut it into halves, either the left hemisphere or the right hemisphere, and then dissociate and enrich for EGFR positive cells from either the left hemisphere or the right hemisphere, EGFR being a marker of GBM for these particular patient-derived xenografts. What this leads to is 54 populations from GBM6 and 18 populations from GBM12, and allows us to compare the tumor cells to the non-tumor cells and also the disseminated tumor cells to the tumor cells from the primary or the core tumor. One of the big challenges here is that when we look at tumor cells from the distal hemisphere, we're getting on the order of tens of thousands or just thousands of cells, of tumor cells from that distal hemisphere. And we really want to be able to characterize this small amount of cells from that opposing hemisphere. All right, as we start analyzing the data, what we see looking at either the proteome or the phosphoproteome of tumor versus non-tumor is that the tumor cells are basically lit up for EGFR signaling, as you might imagine. The non-tumor cells have a lot of sort of classic markers of brain biology, including a lot of oligodendrocyte precursor and oligodendrocyte markers. We can take the phosphoproteins that are enriched in the tumor and then load those into the kinase library and predict which kinases are active in the primary tumor. And one of the ones that comes out is EGFR, as you might expect, as well as a bunch of cyclin-dependent kinases and casein kinases and a variety of other things that are known to be involved in tumor progression. So it gets a lot more interesting when we look at the disseminated cells versus the primary tumor cells, because sort of for the first time, we can start actually characterizing the signaling networks in these disseminated cells. And so one of the things that you might notice looking at the protein or phosphoproteins is that the disseminated tumor cells aren't that much different from the primary tumor cells. They're still tumor cells. They've just crawled away from the tumor and are now in that surrounded by neurons. So even though they're fairly similar, there are some differences, and we can start looking at what those differences are by using gene set enrichment analysis, for instance. And what we see is actually upregulation of neuronal systems, neuronal signaling, transmission across chemical synapses. Basically, these tumor cells, as they crawl away from the primary tumor and go into the normal brain, upregulate processes so that they're able to communicate with the neurons. And this neuronal communication can also be seen here as we look at particular phosphorylation sites that are enriched. And what we see is, for instance, this GRIA2, which is a glutamate receptor. You can see the protein level doesn't change, but the phosphorylation level goes up 5-fold. It's much more active in the background of the neurons in the contralateral hemisphere than it is in the primary tumor. If we look at CAM kinase 2 or this sodium potassium transporter, again, we see that the protein level hasn't changed, but the phosphorylation level has gone up significantly, indicating that these proteins are actually much more active in these disseminated cells compared to the primary tumor. Interestingly, we can go in and characterize this as well by immunohistochemistry. So here we have an antibody that recognizes this tyrosine phosphorylation site on the GRIA2 glutamate receptor. And you can see by these little green dots here that we're getting these sort of punctate foci on the tumor cells. If we quantify the punctate foci on the tumor core versus the tumor margin versus the left hemisphere, you can see an increase in the number of punctate foci as the tumors start crawling away. So one of the other sort of interesting findings that came out of this is this protein called hornerin. And it's actually a pretty poorly characterized protein. There's only a couple of dozen papers on it. But hornerin is upregulated about 7-fold in the disseminated cells compared to the primary tumor. And then it has multiple phosphorylation sites that are all upregulated at least 2-fold, up to 4-fold in the disseminated cells compared to the primary tumor. And so one of the questions that we asked is, what is hornerin doing in these disseminated cells? Because it was one of the strongest hits that came out of our analysis. And so to figure that out, we did a short hairpin knockdown of hornerin in GBM6. And you can see we used 2 different short hairpins here. We're able to effectively knock down hornerin levels. That also knocks down invasion of these cells in an in vitro assay. We can do the same thing on LN229, which is a classic GBM cell line. We can knock down hornerin. And again, that leads to a decrease in invasion. What's actually really encouraging here is we then take these knockdown cells from GBM6, we inject them into the right hemisphere, and then we look at how the tumor grows over time. And you can see that our tumor volume has dropped by about 80% when we knock down hornerin. And I think this is due to the fact that the tumors are not able to invade and push away that normal brain, and so are really limited in their growth potential to be in that one spot. And so through this characterization of the disseminated cells, we can now start highlighting particular kinases that are active in the disseminated cells compared to the primary tumor. And we can start characterizing what makes these cells disseminate compared to the primary tumor, including, not surprisingly, things like more mesenchymal cells. They've upregulated neuronal precursor-like signals. And the primary tumor is really driven predominantly by cell cycle regulation and proliferation, because these cells are rapidly growing, where the cells that have invaded are more migratory and invasive. All right, so the cool thing about this approach is that not only can we look at these disseminated cells, but we can also start quantifying the treatment response in those cells. And so, as most of you know, a lot of the drugs don't cross the blood-brain barrier very effectively. And so one of the challenges has been that we're typically looking at the tumor, but not necessarily characterizing these disseminated cells that lead to recurrence. With this approach, we can now quantify how these disseminated cells are responding to different therapies, including temozolomide. We can also look at treatment response across basically any tumor type. So looking at not just the tumor, but also how the various treatment response might affect tumor cells compared to immune cells or neurons. And then lastly, we're starting to apply this approach to breast cancer metastasis, where we can start isolating and then quantifying and characterizing those metastatic cells in the lungs or liver following breast cancer tumor formation. All right, so in the last couple of minutes here, I just wanted to talk about the other aspect of GBM that's been particularly challenging, which is the immunosuppressive nature of GBM driven by these glioma-associated macrophages. These GAMs comprise about 30% of human tumors, and they drive tumor progression, therapeutic resistance, and immunosuppression. Ideally, what we'd like to be able to do is to target these innate immune cells and wipe out some of these GAMs, which would lead to slower progression, better therapeutic response, and also immune response rather than immunosuppression. And so one of the ways that we've been doing this has been driven by Yufei Cui. And Yufei is a very talented grad student in the lab, and she's been looking at MHC peptide presentation on the tumor cells or on the macrophages, and then seeing how these MHC peptides, which are presented to the immune system and then signal to the immune system to attack the tumor, for instance, how these MHC peptides change as the tumor and macrophages co-evolve together. And so we can take the tumor cells by themselves, or we can take the macrophages by themselves and analyze their MHC peptides, or we can put those tumor cells and macrophages together, let them co-evolve for a few days, and then analyze how they have changed on their surface expression. And ideally, what we're looking for, this is the worst acronym ever, by the way, Yufei, are co-culture-induced very significantly peptides, or CIVIS peptides. And so we're looking for these peptides that are upregulated by co-culture. And the first thing she's done is to sort of characterize that when the macrophages interact with the tumor cells, they change their state and they become more TAM-like, or tumor-associated macrophage-like, when they're interacting with these tumor cells. And so they upregulate various signals associated with sort of an anti-inflammatory response. And then if we look at the immunopeptidome on these macrophages, one of the interesting things is that as the macrophages interact with the tumor cells, they change what is being presented to the immune system on the surface of these macrophages. And there's multiple peptides that increase in response to co-culture with either CT2A, which is a more aggressive syngeneic cell line for GBM, or GL261, a less aggressive syngeneic cell line for GBM. We get, in both cases, we get upregulation of MHC peptides on the macrophages that are associated with cytokine signaling in the immune system and interferons and interleukin signaling, sorry. Yufei's then gone in and quantified these peptides using an approach that we call SureQuant MHC. This allows us to go in and get accurate quantification of these peptides, and not only quantification, but actually copies per cell. So we can now start identifying whether or not these peptides might be targets for either bispecific T-cell engagers, where you're in the thousands of counts per cell, or maybe TCR T cells or CAR T cells when you're in the tens of copies per cell. And so using this information, we can start identifying sort of the treatment modality that we might want to use to target some of these macrophages after they've interacted with tumor cells. And then she's also gone in to quantify basically how the tumor cells are changing when they interact with macrophages. And what's perhaps more surprising is that the tumor cells respond more strongly to this interaction with macrophages than the macrophages do with the tumor cells. And so what we see is actually a pretty strong upregulation of a variety of different peptides after these tumor cells have interacted with macrophages. And what's being driven here is this signaling by Rho GTPases. Rho GTPases basically drive migration and invasion. And what's interesting is as these cells are interacting with the macrophages, we're getting upregulation of these processes associated with migration and invasion. So we can again go in and quantify selected of these peptides by our short quant MHC method. We get up to a 20-fold upregulation of particular peptides after they've interacted with the macrophages. And these are actually the ones that we want to start targeting because these are the peptides in the tumor that have been upregulated by their interaction with macrophages. Okay, so can we start targeting those peptides? What Yufei did next was to identify a handful of these peptides that were presented on either our CIVIS peptides, sorry, that were identified on either the tumor or the macrophages. And then in collaboration with Daryl Irvin's lab, generated an mRNA vaccine to try and target get the immune system to target these peptides. And so using an mRNA vaccine injected on day 1 or day 1 and day 15, or injected on day 8 and day 15. So we tried 2 different methods of trying to stimulate the immune system to attack these tumors. In both cases, what we see is a nice increase in ELISpot formation. We get a nice infiltration of T cells into the tumor. And it looks like we're able to generate an immune response against these particular peptides. Unfortunately, this leads to a very slight improvement in our slight decrease in tumor growth rates, but no overall improvement in survival. And so one of the things that Yufei is working on now is trying to understand why these T cells, when they infiltrate into the tumor, are not necessarily reacting to try and kill the tumor. Are they becoming exhausted, or what is the mechanism by which they're basically failing to attack the tumor? Perhaps the most exciting thing about some of this work is that we've now gone in to look at human tumors, and in collaboration with Kenny Yu at Memorial Sloan Kettering, he's resected a couple of patients now and then taken that tumor and divided it up into CD45 positive macrophages or CD45 negative tumor cells. And what we see is that the CD45 positive macrophages actually have signaling by interleukins and cytokine signaling upregulated on their cell surface, whereas the tumor cells have Rho GTPase and signaling by Rho GTPase. And in some cases, we actually see the same source proteins represented on our mouse model as we do on these human tumors, which suggests that if we can figure out a way to actually tackle and target these particular MHC peptides, then we actually might have a way of moving some of these forward into human tumors. So this would give us a way of trying to basically eliminate the macrophages and attack the tumors at the same time. Okay, so this work with the work I talked about today was really done by 2 really talented people in the group, Yufei Hui and Yijin An. The rest of the White Lab shown here provides awesome support and a lot of other really cool projects. Most of this work was done in collaboration with Jan Sarkaria, and it's been supported by a variety of funds, but especially the NCI Cancer Systems Biology Consortium. And lastly, I'll just give another shout out to Isadora, who helps with everything associated with our group and helped to organize the symposium today. Cool. Any questions?
I have a question on the any plans to actually look at sort of imaging lattices, so use the mouse in the 7T or 14T and look at DTI, DSI imaging and or angiogenesis, because eventually you'll need that to put the models up. right?
That's a good question. So sorry,
So basically, imaging supported, tissue lattices that you need for computational modeling later on if you want to go to discrete, because that's what you need to actually go to these
that's
single cell models.
Yeah, it's a great question. And so we've started working with Natalie Agar using her CySIF, her cyclic immunofluorescence platform, to try and quantify the spatial localization of different cell types in not only in the tumor, but also in the tumor microenvironment and in surrounding these disseminated tumor cells, to try and understand sort of how they're being supported, like you said, with endothelial cells or with vasculature. One of the interesting things that I didn't get a chance to talk about today is that when we see these disseminated cells, if we see them as either individual cells or as a small cluster of cells, their phenotype and some of the characterization is changed between the single cells and the small clusters of cells. For instance, single cells have really downregulated most of their EGFR, whereas as they start growing in a cluster, we start seeing EGFR levels come back up, which is kind of fascinating, but it makes sense. As they go into more mesenchymal state, they downregulate EGFR, but then as they start to proliferate, EGFR comes up. And so I think combining some of our work here to identify potential targets and then look at them by imaging is really a critical component. Oh.
You're the guest. Great stuff. I had two questions. One that actually just came up from your last response, which is that you said that the clusters of cells had a different phenotype than the single cells. Have you looked at associations with immune cells in those cells, and are they different? And then related to that, I don't know if I misunderstood it or if the slide was labeled incorrectly, but on your slide where you're showing data, CD45 positive versus negative tumor cells, what you said was CD45 positive and negative was reversed. And either way, it's really interesting data, but I'm just curious.
Yeah, no, great question. So we haven't gone deep enough into the imaging yet to really start characterizing the immune cells around these disseminated tumor cells. But I think that'll be fascinating to try and get a deeper understanding of what's happening there. On that slide, it was actually reversed. So the CD45 positive cells have Rho-G-DPase, and the CD45 positive cells have tumor. Our tumor cell, CD45 positive cells are the macrophages, and those have upregulated interleukin signaling and cytokine signaling, and the CD45 negative tumor cells have Rho-G-DPase. But the slide, yeah.
GL261, the vaccination experiment,
Yep.
where you did not actually see increased survivorship, and you thought that maybe the T cells are exhausted, but you're looking at that. I guess the surprise is because that model is pretty immunogenic. You can get survivorship fairly easily. So I'm just wondering. So I think that may give us an opportunity to really look at it. Immune evasion in a model that should be fairly immunogenic. And the question: do you think it's just T cell exhaustion, or
yeah, I think
could it be something that's changing with the myeloid suppressor cell population?
And it's a really good question because I thought we'd be able to do something with GL261, and unfortunately, we're not. And I think the big question that comes out of this is the antigen selection process. And so one of the things that we've been chatting with a variety of different immunologists about is once we start identifying these MHC peptides and we're thinking about making a vaccine against them, should we be picking the peptides that are very abundantly expressed? Should we be picking peptides that are sort of in the middle, or should we be aiming for some peptides that are very low abundance? And the difference being that as the T cells come into the tumor, in this case, we pick peptides that were pretty abundant. As the T cells come into the tumor, they see target everywhere. And that's a signal for them that this could be self, and therefore they would get exhausted pretty quickly. On the other hand, if we just go for super low abundance targets, maybe we don't get a response. It's actually pretty challenging to know sort of what's the right range here. And unfortunately, this is a part of immunology that the immunologists tell me they don't know. So it's not like, this is a well-characterized thing. So I think one of the things going forward is trying to figure out, like, as we're targeting these MHCs, which range do we go for, and then which particular peptides are the right peptides to target. And then we haven't been combining this with anti-PD-1 or some of the other checkpoint inhibitors, but we could try activating the T cells with a variety of different mechanisms here.
Okay, so we have 2 comments online. Rakesh Jain, hi Rakesh, has a comment. Another mechanism GBM cells use to migrate to the contralateral hemisphere is a paper that says that there's a glial signature in Wnt7 signaling that regulates glioma vascular interactions in the tumor microenvironment, a Cancer Cell paper.
Yeah, cool. Thank you.
And then we have a question from Divya Sinya, who's asking, sorry if I missed this, does the TAM and or TME downregulate MHC1, thereby inhibiting this cytolytic activity downstream?
Yes, that's a great question. In our data so far, we have not been seeing significant downregulation of MHC1 in vivo. That doesn't mean it doesn't happen. It just means in our system so far, these cells don't particularly downregulate MHC1 in vivo.
Excellent. And then if I can ask another question. So you showed great data co-culturing the tumor cells with macrophages. What about co-culturing more widely with different subsets and frequencies of different cell populations? So not just macrophages and tumors.
Yeah, it's an awesome question. I think moving forward, adding in dendritic cells, adding in T cells, trying to make a more complex multicultural system, if you will be important, but also very challenging. It's challenging because some of those cells are adherent, some of them are not adherent. It's interesting, making a diverse multicultural system like that in vivo is sort of automatic, but doing it in plates in vitro is actually much more challenging than I thought it would be. And also, one of the biggest challenges is those cells tend to grow at different rates. And so as we, even with the tumor cells and the macrophages, as we add those two together, the tumor cells continue to divide, the macrophages don't. And so over 3 days, as the tumor cells grow out and the macrophages don't, we basically shift our tumor to macrophage ratio. And this would be similar if we had DCs or T cells or anything else. And so we have to figure out a little bit, like, what are we aiming for and where do we start in order to get to that point?
And also the spatial localization then changes, right? And who knows what that influences downstream. Do we have any other questions? Okay.
Cool.
And thank you for it. All right, next we have our first